Good afternoon from beautiful Barcelona. I am Alexis Rod, CEO of SciTech Diplo Hub, the Barcelona Science and Technology Diplomacy Hub. Thank you so much for joining one more week of our SciTech Talks, the series of weekly free online lectures that SciTech Diplo Hub and eBay have put forward, together with other world-class institutions and leading international experts in science, diplomacy, and global affairs. Last week, we discussed the future of climate action and environmental science diplomacy after the COVID pandemic. Today, we will address the issue of genome editing, its global governance, bioethical implications, and the role of science diplomacy. The scientific capabilities have advanced so far that the technical ability to edit the genome of all species has already arrived. Gene editing has raised tough ethical questions for as long as the idea has been around, even trickling into popular culture through the movie Gattaca and some episodes of Star Trek and other sci-fi series. But recent events have brought the ethics of germline gene editing to the forefront of public discourse, in particular thanks to the revelation in November 2018 that the Chinese scientist He Jiankui used CRISPR to edit the genomes of twin girls to make them immune to HIV. Around 40 countries have already discouraged or banned research on germline editing, including 15 nations in Western Europe because of ethical and safety concerns. There is also an international effort led by the US, the UK, and China to harmonize regulation of the application of genome editing technologies. However, lack of harmonization is a major hurdle for the commercialization of biotechnology products globally, and this has resulted in an environment where non-tariff barriers, lack of evidence-based policy change, misinformed public dialogue, and expensive uptake of technology has prevailed, all of which are almost certainly contributing to food insecurity, among other negative consequences. In this context, science diplomacy is called to play a crucial role in the discussion on research, bioethics, and regulation of genome editing worldwide. Given the wide-ranging applications in different sectors, it might be necessary to develop coherent governance frameworks which can adapt in line with new technological developments. However, what is the state of the art in the emerging global governance of human gene editing? Can countries develop governance frameworks and arrive at commonly agreed principles? How can science diplomacy help build global common interest on the applications of human gene editing? To shed light on some of these issues, today we are delighted to welcome 4 world-class experts in genomics, bioethics, and science advice, who will be moderated by Emma Martinez, Public Engagement Officer at the Babraham Institute and Project Officer for Open Experiments and Co-Creation in the Orion Project. Good afternoon, Emma. The floor is yours.
Good afternoon, Alexis. Thank you very much. It is my pleasure to be chairing this interesting panel today. So, as you said, now it's really relevant and very timely because newest developments in genome editing technology have led us to a point of no return. Like things are much, the new techniques that exist right now are much more efficient, faster, and they are cheaper as well. Therefore, they have led to a wide uptake of techniques in research labs around the world. So it is really timely that we're having this discussion here. I look forward to discuss with all the panelists, which I'm going to introduce right now. So first of all, we have Luis Montoliu. Luis is research scientist at the National Center for Biotechnology in Madrid and president of the Association for Responsible Research and Innovation in Genome Editing, Enrique. Hola, Luis, bienvenido.
Hello.
Hola, Luis. Gracias. Then we have Hervé Schneeweiss. He is research director at the French National Center for Scientific Research and member of the World Health Organization Expert Advisory Committee on Human Genome Editing. Salut, Hervé, bienvenue.
Hello, bonjour.
We have Julian Kinderlehrer. He is Emeritus Professor of Intellectual Property Law at the University of Cape Town and President of the European Group of Ethics in Science and New Technology from 2010 to 2016 and member since 2005 to today. Hello, Julian. Welcome.
Since he's struggling to connect, but he's getting back.
Yeah, so we will have him here soon, hopefully. And last but not least, we have Shara Chan. She is Chancellor Fellow in Ethics and Science Communicator in the Usher Institute at the University of Edinburgh. Hello, Sarah. Welcome.
Thank you, Emma. Great to be here.
Nice. So we're going to kick off this webinar by starting a round of questions for all our panelists. I would like to start with a question for Luis. So genome editing technologies such as CRISPR have now matured and provided unprecedented opportunities for editing the genome of numerous organisms, ranging from bacteria to primates, which on itself has great potential for medical sciences. The first human trials of CRISPR-based cell therapies are currently underway in areas like hemophilia, sickle cell disease, and some types of cancer. So, Luis, what is the current state of the art in biomedical research in genome editing, and particularly in making edits that will be inherited by the offspring? And how are these technologies currently regulated around the world?
Thank you, Emma. Thank you, Alexis, for the invitation. So genome editing has the capacity to edit any gene of our will in any living organism. And this is something we learned about 8.5 years ago, beginning of 2013, after the proposal made by Emmanuel Charpentier and Jennifer Doudna in the summer of 2012. And for this proposal, they were awarded the Nobel Prize in Chemistry last year. So in the last 8 years, we have learned that applying these genome editing tools enable us to modify at will any gene that we can think of. This has several implications, and there are a variety of applications. This is no longer future. This is already a reality in biology, in the academic world, in most laboratories around the world. So CRISPR genome editing, particularly with these CRISPR tools coming from bacteria, it is a reality, and this is already being used for increasing our knowledge of our cells, of animal models, of plant models. It is also a reality in biotechnology, both in plants and animals, only prevented the applications due to some legislations, which might be a bit more restrictive here in the European Union than in other parts of the world. But perhaps today, what is most interesting for this panel is what is going on regarding the applications in biomedicine. Of course, when we think about biomedicine and we think about genome editing, we think about human gene therapy. And gene therapy, we have to consider whether we will be treating or we will be updating genes through the germline. So that means making this change heritable, or whether we will be treating a human being that is suffering from a congenital disease, and that is normally recognized and called as a somatic gene therapy. Somatic gene therapy is mostly approved and without many problems around the world, whereas germline gene therapy is prohibited in most countries around the world. There are some exceptions that need to be taken into account. For instance, in countries like Belgium, Italy, and Colombia, we have some undeterminations in Ukraine and Burkina Faso, and we have lots of countries that have not decided on this fact. We know that in November 2018, a Chinese scientist decided to cross a couple of red lines, and he was editing some human embryos with the hope that they will be raising kids that will be resistant to the infection from the HIV that is associated with the AIDS. And he was failing. He was convincing some colleagues to implant these embryos, and as a result, three girls were eventually born. These are the three first human beings whose genome has been edited. Of course, none of these girls had the planet modification. It was a very risky experiment. It was a highly irresponsible experiment, because at the stage in which we're living in, the applications of these genome editing tools, at least the first generation of these genome editing tools, has still a number of issues and uncertainties. Not only that we can end up regulating, or we can end up updating or inactivating genes that we were not planned, and these are the so-called off-target effects. Plus, we are not in control of what is going on in our desired gene in the target place. But having this germline discussed aside, what happens? What do we know today regarding somatic gene therapy? So we're checking the database on clinical trials. We will see that there are 46 clinical trials currently using CRISPR tools. Most of them, they are applying ex vivo situations. Ex vivo, in which cells are collected from a patient, and then outside the patient, these cells are edited, and upon confirm and with the expected modifications, the cells are returned to patient, hopefully with a therapeutical impact. And examples of this, as Emma was pointing out, is the sickle cell anemia and the beta thalassemia, not hemophilia. So unfortunately, it's beta thalassemia and sickle cell anemia, plus some refractory cancers that are very aggressive, including melanoma, myeloma, and sarcoma. In all these different cases, we are using a very basic function of this genome editing tool. We are basically inactivating genes. We're using this technique to kill the function of a gene, whereas there is another possibility that we hope, and that we always had hoped, that it would be eventually the champion of these techniques, which is substituting, which is repairing the mutations. And this is mediated by a template DNA that is provided to a system. But still, this is a very challenging method, and it still has a number of uncertainties and issues. I like to say in this couple of minutes that I have left in this short introduction, that there is a new wave of CRISPR variants that we need to take into account. Most of the discussion that is going on nowadays worldwide refers to this first generation of CRISPR tools, in which there is lots of issues and lots of uncertainties. But there are second generation CRISPR tools, including those that are associated with nucleases that are no longer cutting the double strand of the DNA. They are only cutting one strand, and therefore, because they're only cutting one strand, they are not associated with these mosaicisms, with this genetic variability. I'm referring, for instance, to the base editors. Base editors are tools that were envisaged by David Liu in the Broad Institute, and this year, only this year, 2021, we just heard three very neat examples. In January, an animal model of progeria, this accelerated aging. Last month, we heard about the model for reducing the cholesterol levels in plasma. And just yesterday, there was a new study in which base editors were also applied very talently for treating sickle cell anemia. So base editors probably will be the next frontier, and probably the therapeutic tool that we will have to consider. And the ethical issues associated with base editors might be different to the standard genome editing tools, because they have reduced uncertainties and they have reduced issues. 15 seconds just to highlight that besides base editors, we have the epigenetic modifying tools. So we can associate these inactive nucleases to domains that can reactivate genes or can put some genes to sleep. And by doing this, we can also hope to have some therapeutic applications. And that's all. Thank you, Emma.
Thank you very much for the update. Really useful. We will move to a question for Hervé. Hervé, you have led several consensus reports and committees fostering responsible research with genome editing technologies, namely the World Health Organization Expert Advisory Committee on Human Genome Editing and the Association for Responsible Research and Innovation in Genome Editing, ARIGE. So how are these advisory mechanisms articulated, and what should be the main principles driving the governance of genome editing? The floor is yours, Herbert.
Thank you very much. I think that we have first to map the field, because whatever you are working with a non-governmental organization like ARIGE or at WHO, which is a UN agency, you need to design a framework, and the framework must be in the real life, which means that good governance of human genome editing depends upon context. And Luis just described how fast is moving is the field. So you have to consider the context. You have to consider institutions at the national, regional, and global level. And it will necessitate addressing different national capacity. Luis again just named various examples. And the capacity to undertake the oversight and regulation of human genome editing. It will include activities that can be undertaken by UN bodies, for example, WHO, but that will need to be supported by governments. So we need to raise some global consensus. And we need to promote public confidence by ensuring that choices are made in a way that is transparent and inclusive, and involve means to hold policymakers accountable to their choice. So you see that such governance is really something to do. So we need to have some ethical values and principles. And the first set of values and principles are how decisions are made. How decisions are made means that in the world where we are, obviously openness, transparency, honesty, and accountability are basic. Basic for scientists, basic for physicians, basic for the public. We need to develop a responsible regulatory stewardship. And in the same time, we need to develop a responsible stewardship of science. The Montoliu example is just a failure of this responsible stewardship of science. And we need also to manage a responsible stewardship of resources, because there is a cost for research, a cost for treatments. So what are the decisions? We have also to inform what decisions are made. So we need a set of principles. Once again, I think that inclusiveness is the main one. And if we want to raise a consensus, we need to gather for the actors of this consensus. We need to be cautious. And for example, if we want to edit some embryonic material, if we want to move some days to some heritable human genome editing, we need to have the confidence into the technique. We need to have a safe technology. And we even don't know today how to develop, how to assess such a technology at the embryonic level. We need fairness. We need social justice, because these techniques are not only for laboratories. We need non-discrimination, equal moral worth, respect for person. We need solidarity. So you see that making a governance is something to do. And then, only then, you are going to think about laws, declaration, treaties. You are going to think about the peoples that are going to be in charge of doing so. And these peoples are going to be diverse, because you need some regulation of science and some regulation that will be at the governmental level. And obviously, you need to raise awareness in the society. And for that, you need also dialogue, education, and training. So this is why we finish at the end for the WHO report with 8 recommendations, and the report will be soon in the air, because it was already presented to the WHO DG, and it's now under edition. So you need some kind of leadership. And obviously, because we were committed, we were gathered by WHO, we proposed that WHO should take the leadership. But by leadership, we mean the responsibility to gather all the responsible bodies, which mean all the other UN bodies, for example, UNESCO, but also we will speak about intellectual property, so WIPO. You need also to gather the professional actors, and you need to gather the public. So WHO could take the lead to set up some mechanism to promote these meetings and to try to raise consensus among the actors. Then, obviously, and the COVID crisis is an example, you need international collaboration for an effective governance and oversight, because you need to review and you need to oversight. You need to create bodies and instances to review and to promote the oversight. So clearly, it means that everything will need to go through multiple levels. There is no magic bullet. There is no single path. There is no single process. You need to envisage multiple scale and multiple processes. You need to see it at the economical level, scientific level, ethical level. So you need something, once again, global, which means that we are going to deliver a report, 500 pages or more, because obviously I just give you the take-home messages, but you have arguments in our report. And clearly, this committee will not be enough for others to oversee what is going to happen in the future. So WHO and the international community will be in charge, for example, with the national academies, to create a successor to our committee to review and to oversee what is going to happen. We tried already some mechanism. For example, we already set up a human genome editing registry. It was based on the existing ICTRP registry existing at WHO, which is the equivalent of clinicaltrial.gov. It's clear that from the pilot studies that we made, this registry failed to recover the keywords, the correct keywords, and to really find the studies, clinical or preclinical, that could lead to some John QA story another day. So we need to promote new registries for research and for clinical applications, and we need to develop that. We need to take into consideration the fact that the world after COVID will allow traveling, and how to prevent ethical dumping. One scientist not allowed in one country moving to another country. So we need to raise an international consensus that we need basic, once again, the basic recommendation is in any country you need a review, an oversight mechanism, and if a country has not this regulation, it should be forbidden to do any kind of human genome editing in such country. And then you need to fight again what we see already existing in the field of stem cells: illegal, unregistered, unethical, and unsafe research or applications. We have plenty of raw clinics for stem cells. It should be terrible to have plenty of raw clinics in the field of human genome editing. So once again, it's a multi-scale and global story to prevent these illegal, unregistered, or unethical research or application. We also need to face the reality of cost and development. Luis spoke about beta thalassemia or sickle cell anemia. How these treatments are going to reach the people that need this treatment. So you have a question of intellectual property, and you have the questions also of infrastructures to produce the treatment. And in these conditions, WHO should gather other organizations in charge, such as WIPO, and discuss these questions. And finally,
it is, if I might interrupt here, Herbert, it is clearly from your answer that it's highly complex to develop such a governance mechanism. And I am aware that it has taken quite some time to develop this governance framework that you are discussing about. But if you allow me, we will move on into the next question, which, because now we have already touched into the safety and efficacy of the technology itself, and also with you in the governance frameworks. So I would like to move towards the ethical issues surrounding the gene therapy. So how are considerations about autonomy, privacy, choice, and fairness balanced? And should we agree, decide, and act together? And if so, how? And I think this is a question which is very appropriate for Julian, if you would like to take the floor now.
Hello, hopefully you can hear me. Good evening, everybody. The European Group on Ethics, of which I've been a member for an enormously long time, has produced an opinion on the ethics of genome editing, and it addresses the profound ethical questions that arise when editing modifies the genomes of higher organisms. It addresses humans, animals, and plants. So I'm really going to talk a little bit about what that opinion has done, because it's published. It was published in March. We saw safety as the major ethical issue, I'm afraid. And interestingly, we said it was an ethical issue. And I'll put a number of issues within that. And that's not me. Playing God was one of the things which we had to address. Playing God by interfering with the genetic material. Is this only for germline modification? Or would it apply to interventions where modification of the germline was not intended? If the latter, all medical procedures would be suspect, because any medical procedure involves the modification of the human being. And some religious sects assert that you should not use medicine at all. Different societies will have a different take on this. Could different societies have different laws because of this? And should we accept that the difference, say, between South Africa and Ubuntu, and a much more rigorous legal system such as France, could address the problems differently? One ethical standpoint could be that editing of the human genome is never acceptable, regardless of whether it is in the germline or somatic. And I feel that the EGE actually said it is acceptable given many conditions. We have to accept that all medical interventions have a degree of risk. Risk and benefit calculations are essentially made by physicians, but patients should be given enough information to make informed choices. If the intervention is characterized as research, it needs ethical approval. If it is characterized as an established medical procedure, it doesn't require ethical approval, it simply requires informed consent of the patient. So we've got to be clear as to what the issues are in virtually all jurisdictions. It's not as simple as we would like. What constitutes safe enough? See, all medical procedures have risks and benefits. What exactly in this case is safe enough? And it's a trivial issue, and it is clearly an ethical issue. The proportionality of benefit in terms of preventing a serious genetically transmitted disorder must be balanced with the risk of not only failing to correct that defect, but also of introducing unintentional modifications that could have serious implications for future generations. And we have to think about that. One extra thing which I wanted to, well, two actually. One was we could modify animals to provide humanized tissues for transplantation into humans. And the pig has been mentioned for that. Does this have implications for the humanness of the individual into which such tissues are placed? Governance is likely to be different in jurisdictions that have different traditions. We should take note of that. We can't have a single system for the world. Diversity. Could and should genome editing diminish diversity amongst human populations? Diversity is an accepted good and a social goal to be protected and promoted, often against the background of its perceived precariousness due to human activity. So we've got to be very careful there. Congenital deafness was one that came up while we were talking about it. And some parents said, we want our children to be deaf, even if you can correct it. How should we deal with a problem like that if this technology allows us to modify it? There's much more. Somatic cells should only impact the recipient of the treatment. Should this technology be available even where treatment short of genome editing exists for treating the condition or alleviating its symptoms? Can we use a different technology? And if so, should we use this technology? Modification of the germline requires permission of the recipient and any offspring. How do we do that? Can we say that? Let's take examples. Tay-Sachs, where the infant dies immediately on birth virtually. Would somebody who was likely to have Tay-Sachs approve the fact that they might live a bit longer? Or we can move to Huntington's disease, where no treatment exists, but for most it is a late onset disease. And Woody Guthrie, one of my favorite musicians, was a sufferer with Huntington's disease. We have to think about what we're using this technology for: enhancement, prevention, therapy. Whilst many consider therapeutic modification of human cells to be acceptable when it is safe and where the individual is given consent, should enhancement of normal human beings be allowed? Ian Thorpe, a swimmer, had size 16 feet, and people talked about him as having flippers. There is one teenage boy who is in England who has size 19 feet. Will he be a Olympic swimmer taking after Thorpe? Should we even allow modification of the human genome to enhance people to this effect? Where is the boundary between therapeutic use, prevention, and enhancement? Is there an ethical divide between these, which indicates that only therapy should be permitted, if at all? And can we be sure that somatic modification does not modify the genome? There's things about naturalness. This technology may be expensive. Should it only be available for those who can afford it? And I think that has been mentioned already by Hervé. International agreement is necessary to ensure justice for all, but we have to find a way to do that. Finally, if I may quickly, many people have talked about a moratorium. To me and to our group, a moratorium has to have an endpoint. You have to be able to identify what you want to have achieved to end a moratorium. Otherwise, it's a ban. So what are the conditions under which a moratorium would be waived? If we're talking about that, those are the major issues which the European Group on Ethics, the opinion for which I helped write, has talked about. Thank you.
Thank you very much, Julian. It was really interesting. And I take your point on diversity, and I extend it to any other species, not only the human one, if we go on to editing the genomes. So my last question will be now for Sarah Chan. So Sarah, you have been advocating in the UK for a more forward-looking approach to genome editing. How would you reply to the major objections to genome editing in humans and the so-called precautionary principle, where there is limited scientific evidence, but the stakes are really high? And what will be the main drawbacks of this global moratorium on genome editing that Julian just mentioned? And lastly, how would an appropriate framework for regulating this fast-moving area of science would look like for you?
Thanks, Emma. I don't think I'm going to be able to comprehensively answer those 3 questions in the time available, but I will do my best to offer some thoughts. So I very much agree with Julian in saying that if we're going to say it's a moratorium, we need to be clear about when we can move away from it. Otherwise, it's just an indefinite moratorium is essentially a ban. And in the case of genome editing and heritable human genome editing, I don't think that that is indicated. So when you say I've advocated for a forward-looking approach, that doesn't necessarily mean that I think we should throw out all the regulation we currently have and move to heritable genome editing straight away. But we need to be very clear about what we are basing the moratorium on. So why are we currently hesitant about the idea, and what would need to be satisfied, what sorts of conditions would need to be satisfied in order for us to say it is now ethical to move forward with thinking about whether we want to implement heritable human genome editing. I think one of the main issues that has been raised both historically and in the last few years as different expert groups and committees have considered the prospect of heritable genome editing is obviously the safety and the efficacy of the technique. And you always hear those 2 terms together, safety and efficacy. In other words, does it work? Does it work well enough? Again, I thought Julian put this point very well. Is it safe enough? In other words, is what we know about the safety and the risks and the efficacy and the benefits such that we can say there is a positive balance of benefit to risk. And so I want to break down considering this concern over risk. You know, often people will say, oh, it's too dangerous, it's too risky. Now, is that a knockdown objection? And I'm going to argue that it isn't. What it requires us to consider is too dangerous in comparison to what? So what are the alternatives? It requires us to say, what are the dangers? When you say it's too risky, what are the risks about which we are concerned? What are the potential harms? And then finally, it requires us to ask, what should governance do in the face of risk? So what is the proper governance approach to take when there are both risks and benefits? So let me address those 3 points in turn. When we say it's too risky, we are saying in comparison to all the other possible options we might have. And those would include to do absolutely nothing. And in the case of a family who has a genetic disease, the consequence of do nothing is the harm of that disease in that family's children and potentially in their children's descendants. Other options might be to use somatic genome editing as an alternative. And so many people will argue that because heritable genome editing carries additional risks, and we'll talk about in a moment about what those additional risks might be, that we should stick with somatic genome editing as a safer option. And of course, there are the other alternatives, such as using pre-implantation genetic testing, which has been advocated for a number of diseases. So we have to think in conjunction with all of these alternatives. I think, and this has also a bearing on our concerns about therapy and enhancement, that when we're faced with the near certainty of a very life-limiting genetic disease that's going to cause a huge amount of suffering, both to the child born with it and to their families, we would have to be pretty sure that the risks were equally huge to say it's too risky. And that's why I think that the indications for the application of heritable genome editing in humans will first be most ethically justified where they come about to prevent such disease. If you think by contrast of the consequences of doing nothing for what we might think of as a therapeutic enhancement, the harm, if you like, of doing nothing is that perhaps your child is not Ian Thorpe. I admire Ian Thorpe very much, but there are certainly worse things in the world than not being Ian Thorpe. So that's where I think, rather than having to draw a hard line between therapy and enhancement, we may be able just to think about it in terms of what the relative risks and benefits are of either doing genome editing or not doing genome editing. Moving now to the risks and relative risks and benefits of the other alternatives, somatic genome editing or something like pre-implantation genetic testing. This requires us to say, actually, what are the harms that we're concerned about? And I want to argue here that the potential harms we're concerned about when it comes to heritable genome editing are not qualitatively different to the potential harms implicated with somatic and with somatic genome editing and with PGT. Now, let me run you through that because it seems to many people a counterintuitive argument that the risk of heritable genome editing affecting the genomes of future generations, the genomes of descendants, is obviously a greater risk. And Julian, I'm going to respectfully disagree with you here that actually we do require the consent not only of the person who will be edited, but of their descendants to heritable genome editing. Because of course, every child has probably said to their parents at some time, I didn't ask to be born. And children who are born as the descendants of someone who's genome edited didn't ask to be born, but neither did the children of people who are not genome edited or the children of people who have been born as the result of PGT. No matter how their parents' origins came about, those children will never have had any say in it. Again, if you like, for genetic disease, we might justify heritable genome editing in the same way that we justify fetal or neonatal surgery without consent. It's ethically justified as a matter of necessity to prevent suffering and disease. So I don't think we do actually need the consent of future generations, but that's perhaps something we can bring out in the questions and discussion. Let me go back, though, to what the actual harms are that we're concerned about, what the risks are. So obviously we are concerned that off-target effects might result in introducing some worse mutation that has a harmful effect. But that's the case both in heritable genome editing and in somatic genome editing. The difference, of course, is that these side effects of somatic genome editing are not necessarily passed on to future generations in somatic genome editing. In heritable genome editing, the side effects are only potentially passed on, and it depends on the reproductive options and the choices made by the person who's been genome edited. So if you are someone who has been born as the result of genome editing intended to cure some genetic disease, and you've been born without that disease but with some other consequence of an off-target event, you have the choice to risk passing that on to your descendants, but you also have the choice not to reproduce and to not pass that negative mutation on. Now, that is obviously a very difficult reproductive choice to say, I can have children, but I risk burdening them with a genetic disease. But that's already the choice that is faced by current would-be parents who have genetic disease. So it's only passing the potential of a difficult reproductive decision. It's passing the reproductive risk further down the line. Now, in my mind, there's a very important question as to why we should prioritize the reproductive rights of future people who may or may not want children, who may or may not want the option of passing on a genome without disease. Why would we prioritize their reproductive rights over people who currently do want children and who want not to pass on that disease? Somatic genome editing does also affect the genomes of future generations, even though it doesn't affect them directly, because what it affects is whether people with a genetic disease survive to grow up and to reproduce. So children will exist as a result of somatic genome editing who wouldn't have existed had that procedure not taken place. And then finally, when we think about the costs and risks of heritable versus somatic genome editing, sure, heritable genome editing has the potential that if certain decisions are made, any side effects might be passed on, but those decisions don't have to be made. So we're talking about a single procedure that potentially results in benefits for all of the descendants. So as much as the risks multiply, the benefits of the procedure, if it works, also multiply. Somatic genome editing, on the other hand, you need to repeat in every generation. There are costs and harms of the treatment itself, both to the health system, but also to the patient as they wait for whatever bone marrow harvesting and modification and transplantation to happen. And every time you do that, every time you have to do that in each generation, the risk is multiplied. So at some point, in fact, the risk of those cumulative events is going to exceed the risk borne by a single individual in heritable genome editing. So my argument here is that we need to take into account the intergenerational distribution of risk. Finally, sorry, I'm just getting pop-ups in the chat. Yeah, let me conclude with a couple of thoughts about the way this plays out at a global level. I think in terms of governance, we need to be very careful of imposing a high-income country perspective on the evaluation of risk. So when the attempt to genome edit the babies to be immune to HIV became known, there was some discourse about whether our perspective that being immune to HIV actually isn't a benefit worth genome editing for, whether that holds true everywhere. And arguments were made that actually in a situation where HIV is, being HIV positive is more stigmatized, where treatment's less available, you know, our evaluation of being susceptible to HIV really isn't that bad is framed by our assumptions about the society we live in and the treatments available to us. Now, I'm not arguing in any way that what was done in that experiment was right. I think there are many reasons why it's not right. But just to say that we need to take into account socioculturally relative differences in how we think about risk. We need to think about our assumptions about the alternative options and whether they're available. So PGT is not available everywhere. There are serious resource considerations of implementing something like a national screening program or a national somatic gene therapy program for some of the diseases, sickle cell anemia was mentioned earlier, diseases that pose serious health burdens in low and middle income countries. Now, we need to be realistic as to whether it would be feasible and possible to deploy genome editing technologies in low and middle income countries to address these. But we need to not assume that all countries will have either the same ethical concerns or the same resources and the same health system availability that we have when we make a determination about which risks are worth taking.
If if you want to say one last line, because I was going to just extend those thoughts to the rest of the panelists.
No, please go ahead. Thanks, Emma.
Okay, thank you very much, Sarah. It was really interesting points you made. So I think very briefly, I would like to ask all the panelists. So how do you think that science diplomacy contribute to, or can contribute to, better global governance of genome editing technology? So already Sarah has comment on this point. So I think it would be very happy if the rest of the panelists would like to add very briefly a line on this.
Okay, Emma, I would say just to follow on what I've been just listening from my fellow colleagues, is that we're facing a moving target. So most of what I hear, it refers to the traditional or to the classical CRISPR-Cas9 genome editing, but we have to admit that we're no longer there. We just move on, and most of the applications that I've been reporting on, that have been published and reported just in this year, 2021, they're using variants that they are no longer associated with indels, they are no longer associated with genetic variability, they're no longer associated with significant off target. So we need to rethink and we need to consider that the technology, it's moving much faster than the discussions that we're currently having on the governance of genome editing. And that is to identify the risk that Julian was referring to. And the risks have changed, and the risk, I'm sure they will continue changing. When we were talking about genome editing a couple of years ago, we all thought about using templates to fix mutations and to substitute the mutant gene by the correct gene. Well, as a practical terms, this is not happening, and what is happening is that the medical opportunities have been found, but just inactivating the mutant gene rather than correcting. And sometimes this has the same or even better therapeutical effects, whereas has much lower risk. And just a couple of lines to say regarding this governance and regarding how are we facing and how we addressing it at the worldwide level. Well, what I learned from lawyers is that there are no international laws. Only thing we have is national laws. At the international level, the only thing we have is declarations, we have recommendations, and then we have to rely on what Hervé has presented us. We have to rely on United Nations, WHO, we have to rely on other agencies that can represent the hope and the aims of the mankind, the human mankind, and we just hope, and we just have to hope that by just updating these declarations, by just introducing some new phrases, some new recommendations in this Human Genome Declaration or similar documents. We hope that this will be permeating and this will be being transposed at the national level. And this is probably the task that we have to be all working together, trying to convince every one of us to the different countries, so that we can sort of harmonize. Probably, as Sarah was just pointing out, it will be very difficult to reach consensus because of this cultural difference, but at least we need to try to harmonize this and to try to balance and to leverage, so that we can all progress and extend these genome editing benefits to everyone. So not to forget this justice, this principle of justice that Hervé was also referring. And I say no more. Thank you.
Hervé or Julian, would you like to add something in this regard?
I think that something that could not be acceptable is to have multiple standards. For example, some standards in Europe and another standard in Africa. This double standard is something that is unbearable, at least in my view. What we need to have, and what we see already for the last 20 or 30 years since the development of assisted medical reproduction or stem cells, is the development of these review and oversight mechanisms. And on the process, we can have a global consensus. We can have, and we see since the 80s that most countries are developing national ethics committee, review boards, oversight principles, that health ministries in every country are developing systems to review and for oversight. So on the global process, we can find mechanisms that obviously will prioritize in some countries for some disease, that will prioritize research, that will prioritize healthcare. But the global process and the development of this process at various scales will be the same. The other thing where we can raise a consensus is education and public engagement, because on one end of the curve you have the scientists that are producing the new knowledge and the moving targets that Luis explained, but on the other side you have the general public, the general citizens. You need education, you need to understand what it means. At this stage, the problem of heritable genome editing is that we don't know how are the processes of DNA repairs or DNA modification at very early stages. So we need just more research on this point. It's just not doable, if whatever is the technique. We are working on somatic. So we need education. And then we need engagement, because we need to know what the peoples really want, what are their priorities. And finally, we need empowerment, which is easy to say in our democratic societies, but a basic need for most societies. So we need to combine all these elements to really develop, and this is true for genome editing, but this is true for any kind of emerging technologies.
I think the point you just have raised, Hervé, is really relevant for science diplomacy, as in this can be something where science diplomacy can really make the impact in terms of training, education, and engagement/dialogue. There we can learn a lot from each other. So, Julian, would you like to add something else?
Yes, please. I actually disagree with Hervé to some extent. And I look at the picture of abortion, even in Europe, and different countries have very different standpoints. We live with it, unfortunately. We can't impose abortion, let's say, in Poland and allow it and not allow it in Britain or vice versa. So we have a problem there. And the same with PGD, the same with assisted suicide. So many of these similar issues aren't governable by an international agreement. I'm sorry, it doesn't work, and it won't work, because nobody in Britain will be prepared to accept that PGD abortion is not allowed. And we would actually, I think most of us, according to the last opinion poll, believe that assisted suicide should be allowed. But that's not yet allowed in Britain, but allowed in France, allowed in Belgium, allowed in the Netherlands. So we have to be aware that the different cultural traditions have to be addressed. But that gives you tourism, medical tourism, if I can call it that, and we have to think about how you handle that. Can I stop somebody from Poland going to France for an abortion? Let's think about those issues.
Thank you very much for raising that important point, Julian. So if you are all fine, I would like to open the floor for the Q&A with the audience. So I will see what questions we have. So the first question that has received more votes, I think it has been addressed, I believe, by Sarah, as in what would the global framework for genome editing would look like, and what will happen with countries that do not participate. I think Sarah has commented briefly on this regard, so I will move on to the second question. Sarah, would you like to add something else, or someone else?
I think that a global framework that doesn't involve the participation of all countries, but especially the major players in the technology, will be, it will not be worth very much, not least because of the problem of research tourism and medical tourism. But I don't think, and I know somebody asked about this in the chat, medical tourism is not necessarily a bad thing, but the governance framework needs to take account of the possibility, in fact, the likelihood of it happening, and of the ethical consequences, for example, in terms of access to technologies and so forth. But I think every effort should be made to engage all countries in a regulatory framework. And that may mean that we cannot have a framework that says it's permissible, it's impermissible, but that's not what a governance framework necessarily needs to do. I think there are principles that could be embedded within a framework that we could hope that all countries would sign up to.
Thank you very much, Sarah. Thank you, Veronica, for that question. So I will briefly move on to a second question, but we will be quite brief. So Felix Moronta asked, conversations are focused on the impacts on humans, but other organisms are intensively being engineered. Manage organisms like crop plants or farm animals, and even wild populations like mosquitoes or rodents. How to better mainstream the biodiversity in the genome editing impact conversation, which I briefly mentioned throughout the session today. So I extended that to all species. Someone wants to answer this question? Implications for all species.
Well, I can say something, Emma. Thank you for the question. Yes, definitely, genome editing goes beyond human beings, and this is already being used in plants, in crops, and in animals, in livestock, and in also being projected, not yet being used, but projected in as a method to control plagues. Well, I have to say that most of these genome editing approaches, they are much more secure and safer than approved applications that we have been relying on for biotechnology over decades. So I think my view is that we have to embrace these new technologies. However, there are in different parts of the world, these technologies, they are not perceived the same way. And we need to remember that we're living in a global world. So if we are preventing from applying these technologies in a given part of the world, simply the companies and the production of crops, of animals, will be moved to other parts of the world where these technologies are permitted.
Thank you, Luis. Someone else would like
to come in as well.
Yes,
the EGE opinion, which was published in March, and you can get it on the web, addresses animals in very great detail, plants in significant detail, and even gene drive, which the mosquito reference would address. So get it on the web, genome editing, EGE, and a document is available freely to anybody who wants it.
If I might ask, if I might add a little bit from my own contribution in order to mainstream the conversation, the dimension of biodiversity in several species in the impacts of genome editing, what I have learned over the last few years, there has been public dialogues and engagement efforts in both the agricultural uses of genome editing and farm animals, or they are being developed at the moment. So it's a way of bringing society along with technological developments. But thank you very much, Felix, for the question. If no one else wants to say anything from our panelists, then I would like to briefly wrap up. So today we have discussed about some of the technological issues around genome editing and the newest developments, possible governance frameworks and approaches, the ethical and societal implications, the need for foresight of technological developments, and the opportunities that all these issues of genome editing bring to science diplomacy. So similarly to a highly infectious virus who knows of no borders, organisms whose genome has been edited, they can also travel across borders. They can breed, and they can introduce unprecedented changes to the status quo. Life as we know it can change, and it is in the hands of all stakeholders to collaborate to support a change for the better. So thank you for the organizers for this fantastic seminar. Thank you to the speakers for putting these thoughts into this interesting topic. And I pass the floor back to Alexis.
Thank you so much, Emma, for this outstanding moderation, and thanks a lot, Luis, Hervé, Julian, and Sarah for this enlightening discussion. It's a pity that we don't have more time. We were actually already out of time, but we have dozens of questions in the Q&A tab. I'm sure that many of them will have the chance to be answered through our social media, so we can follow up the conversation there. And as you were just mentioning, Emma, as we saw today, science diplomacy is emerging as a key tool to empower the voice of science in global affairs while developing better governance mechanisms to navigate this complex and fast-paced world we live in. Cross-border collaboration advances at a fast pace, achieving significant feats of scientific discovery, as it was mentioned today, from the application of CRISPR to treat genetic disease to identifying the SARS coronavirus. So in recent years, researchers have increasingly become transnational actors in public policy and global governance. But we saw that during the current global health crisis, we witnessed that science matters, but that science and the ideas of scholars and scientific researchers also need to be made to matter. And one hopes that countries will seize the opportunities for science diplomacy to play a positive role and give the importance it deserves in this challenging task. I would also like to take this opportunity to remind to our audience that applications for our Science Diplomacy Summer School are now open. This unique 2 weeks university course will be held this July in an online format and organized by SciTech Hub and eBay, together with other leading institutions, will allow you to get certified in science diplomacy and further into some of the topics we have been discussing today, as well as other pressing issues at the intersection of science and diplomacy, including climate change, cybersecurity, AI and tech diplomacy, sustainable development, global health, and the role of global cities and emerging economies. Thank you very much for tuning in today, and please join us again next Thursday at the same time to discuss on the role of science diplomacy in the global south. If you haven't done it yet, don't forget to register. Thank you so much, stay safe, and see you next week.